All right. It looks like it is 12 o'clock on the dot here in Minneapolis, so we will get started. Sorry about that. We're so glad to have you all with us today. Thank you for joining us for the special virtual edition of our Dean's Lunch and Learn service, or excuse me, series. Today we're featuring the Department of Biomedical Engineering. My name is Megan Orr, and I am the event director for the college. And my role today is to moderate the chat as well as facilitate Q&A. If you'd prefer to listen today, you can do so via our live phone option. So if you haven't done so already, you can call 651-372-8299 and type in the webinar ID, which is 975-741-0815. If you're having technical issues, please, again, go ahead and type them into the chat. Today, you're welcome to chat with each other using the chat, but if you have questions for any of the presenters, please type those in the Q&A. That is where those will be monitored. We do have some time set aside at the end of the session for Professor Johnson and for Professor Lim to answer the questions that you've submitted. And we are recording today's webinar, and we'll share the link with everyone who's registered after this event in a follow-up email. It usually takes a day or two for that to go out, but you're welcome to share that recording with others who weren't present today. We'll also have that posted on the CSE YouTube channel for you to watch at a later date or for you to share. And now I am pleased to introduce Dean Mas Kaveh. Mas has served as Dean of the College of Science and Engineering since 2018, and he has held a variety of roles in the college for more than 40 years. Before serving as Dean, he was our Associate Dean for Research and Planning, and prior to that, he was head of the Department of Electrical and Computer Engineering from 1990 to 2005. Dean Kaveh joined the college, joined the university in 1975. Dean Kaveh, you can go ahead and unmute yourself and you can take it from here.
Thank you, Megan, and good afternoon, everybody. Thank you so much for joining us on this 21st century meeting. I am, excuse me, coming to you from the mall right outside my office in Walter Library, as you can see. And whereas we much prefer to have a normal meeting with many of you on campus in Walter Library and be able to enjoy some refreshments and light lunch together with you. I'm sorry that we can't do that exactly, but at the same time, I'm absolutely delighted that through this new medium, we're able to reach so many more of you. As we've already seen, a number of you are actually from the West Coast and East Coast and so forth. So thank you so much for joining us. These Lunch and Learn series is an opportunity for us to connect with our alumni and friends and be able to essentially reacquaint you with our featured departments and the work of some of our distinguished faculty. And of course, today we're delighted that we're doing this with the Department of Biomedical Engineering. I don't have to tell you that we are operating in a rather surreal time. Our staff, students, faculty have all pivoted in many different ways, in ways I assume you're familiar with, ever since last March. And as we speak, we are planning, together with the rest of the university, our onboarding in essence for fall. Much work has been done in our departments under the condition that our first and foremost concern is the safety of our students, our staff, and our faculty. So everything that we've done in planning for essentially resumption of hybrid teaching in the fall, and actually what we've already resumed our laboratory research activities during the summer has been predicated on conditions to making sure that safety is first and foremost. And I say this because while we've done all the plannings, and as I said, many of you must have either read or heard about it, we are going to offer a complement of a mix of classes that are face-to-face, particularly the laboratories in our college. And most of our classes actually delivered online or remotely in different ways or as a mix, to a large extent because, frankly, to again operate under the safety rules, we have to keep the distancing, appropriate distancing satisfied, which does not allow us the space to manage larger classes, frankly, any class larger than 20 perhaps across the university. So these are the things that we've all been working on. We've been working on these while at the same time being very much aware that this unfortunate situation with the pandemic changes on a daily basis. And I'm sure many of you have been reading about all the developments and pivots that many of our other academic institutions have been following, the ones who have already started or are about to start, such as University of North Carolina, that just yesterday pivoted into an online mode of instruction. So we will do our best. We will do certainly our utmost on campus to manage everything safely and appropriately for our students and hope that everything will proceed as we have planned. But the university also very much prepared to continue our instructional activities and give the best education we can if we have to pivot in new modes of instruction, again, beyond what we have planned. So under all of these conditions, what I'm happy to tell you is that our focus on the grand strategic plans for the college continues unabated. We, as you have heard in the past, or you have read about it, we've had the plan to expand the college to again, respond to the demand for our programs and also the workforce demands of the community and particularly industry, both in Minnesota and outside of Minnesota and globally. And we are on track to do that. We may have taken a little bit of a pause from actual growth this fall. We won't know until we actually start the semester, but at least at the moment, we don't see things going in the wrong direction for now, and hope that much sooner than later, we all will be back to relatively normal operations. We're also continuing our commitment to diversify the college, diversify in many, many respects. And I'm happy to tell you that with the incoming class of first-year students, we're already seeing that the demographics of that incoming student has continued to move in the right direction, actually reaching record levels, both in terms of gender in our incoming students, as well as in other representations of underrepresented groups within the College of Science and Engineering. So we're going in the right direction. We're doing that also with faculty, but much more work to be done. Let me take this opportunity to thank all of you. Thank you for your commitment to the college, to your connection, staying connected with the college, and particularly in this very difficult time for so many of you to have contributed to the College of Science and Engineering Response Fund. And this response fund, which now is over $100,000, has been critical for us to be able to provide some needed assistance to our students, and also to deal with some unexpected expenses for the college that are related to the COVID in particular. For example, this summer we've created a large number of physics experiments in the form of kits that have been supported with this response fund. This is a way for us to be able to give our students laboratory experience even when they're not on campus. So these are just some simple examples, but my time is short during this talk, and you're here to actually listen to my colleagues who are with us from biomedical engineering. So let me stop for now. Thank you again, welcome you to this meeting, and introduce my distinguished colleague, Brenda Ogle, the head of the Department of Biomedical Engineering. Brenda became the head of the department in July 2019, and she has been a faculty member in the university since 2013. Prior to that, she had been a faculty member at the University of Wisconsin-Madison and also at the Mayo Clinic. In addition, because Brenda has so much time on her hand, not enough responsibilities, she also has been leading the University of Minnesota Stem Cell Institute, as that institute has been looking now for new leadership as well. So thank you again, Brenda, for everything that you're doing, and I hand it off to you to introduce the department and your colleagues, our colleagues.
Great. Thank you very much for the introduction, Dean Kaveh. And also good afternoon to all of you who are here. I'm so excited and pleased to be joining you today. And as was already expressed, I really wish that we could be together in person. But I think that will happen eventually. And as also was mentioned, we couldn't have all seen each other from across the country and even globe. So this is a unique opportunity. I was grateful to connect with many of you as I took on the role of head this past fall. And I want you to know that we are continuing to plan for more in-person events post-COVID. So please stay tuned. In the meantime, please look to our website and social media. We've been trying very hard to keep it even more updated than usual because we can't be together. And also our biannual newsletter. So in the last one, which I hope you received, I shared with you a summary of some of the discussions, important discussions we've been having as a department about the values of our department and indeed our field. We think about, when you think about BME at least, we think about innovation, creativity, integrity, compassion for others, and service. And the fact that these values demand that we directly engage with the issues that we're facing today, both the long-standing injustices laid bare by George Floyd's murder and also the global pandemic that has really upended, I think, everything in at least my life, and I'm sure all of yours. So I've also, in that newsletter, shared some of our plans to meet these challenges going forward. And I want to extend today an invitation to you to please reach out and share your thoughts, how you think our department can be an influential player in helping the nation and the world engage, move forward, respond to these challenges. So, and then even today you'll hear, as part of our discussions, description of studies, we actually have 7 different faculty members undertaking studies to either diagnose, or understand, or treat patients infected with the COVID virus, and you'll hear some of that today. But we start our conversation with a fascination that I have, and I think many do, with the brain. We're always looking, and have been for centuries, trying to understand how it works, and in so doing, understand exactly what separates us from other life forms, and what we do when it fails. So you may remember that the study of the brain and the nervous system got a pretty big jolt in about 2013, when study of the brain was identified as the next great American project in the form of the Federal Brain Initiatives. Many of you, I'm sure, remember, but what you might not know is that from that Brain Initiative, your tax dollars actually supported the early work of our speakers today, Professor Matt Johnson and Professor Hubert Lim. Their work has resulted in the development of devices that are used to treat a range of brain dysfunctions and disorders, and has supported the creation of the synergistic centers and institutes that you see on this slide. And so I just wanted to share a little bit, just so you understand the magnitude of what's going on at the University of Minnesota and how BME faculty are really leading the charge in many respects. So if you look sort of toward the middle, the Institute for Translational Neuroscience was actually a presidential initiative at the University of Minnesota way back in 2003. So a good 10 years before the federal brain initiative. And part of the dollars from that institute have gone to setting up the Center for Neuroengineering, which actually another one of our faculty, Professor Tay Nedoff, is the director of. In addition, it fueled MnDrive, which is a state-funded initiative to study the brain. And that has also led to just a large nidus at the University of Minnesota, led to the Center for Magnetic Resonance Research in the Department of Radiology, and also a new center that's not listed here for applied and translational sensing science. And those are funded in part by the National Institutes of Health and also some local industrial investment. All of this is pushing therapies toward the clinic in partnership with clinicians from the medical school, mHealth, and also the Earl Bakken Medical Devices Center, which actually Matt Johnson, who you'll hear from today, co-directs. So all of that just to provide the context. The context is that Matt and Hubert have been integral forces in these centers. They have been thought leaders really for the university, local industry, and the world. And one of the concepts that you're going to hear them talk about is something called responsive medicine. So you've heard about precision medicine, or the desire to tailor a therapy to an individual. Responsive medicine takes that one step further. We're talking about engineering therapies that can respond to an individual's reaction to that therapy, and then further in real time adjust the therapy. And so you'll hear some of that today. So hopefully you can hear from my voice how excited I am and incredibly proud of these two colleagues in our department, and the amazing consequence of having their work happen at the University of Minnesota is that the insights gains are exponentially expanded by the students that they train. So I think it's nearly 200 just out of these two labs in the past 10 years. And I'm guessing many of you on the call today are some of these trainees represented here. So I want to end by reiterating Mas's words of thanks for all of you for staying connected and engaged. And before I take up all of the time together, I want to pass it over to Professor Matt Johnson.
Megan, would you be able to allow me to share my screen?
Yes, one moment.
Great. Thanks so much, Brenda. It's a really great synergistic environment in our biomedical engineering department, led by Professor Ogle as the chair, and it's as you'll see, there's so much collaboration going on amongst faculty, and not just faculty in our department, but faculty across the university. So let's see here, I'll do it. So the title for the presentation, at least that I'll provide, is this Road to Responsive Therapies for Damage and Disease of the Nervous System. And between myself and Professor Lim, we're really trying to understand this incredibly complex system that's both complex in anatomy and physiology. And just to kind of give you a sense of the scale of this complexity, we have around 100 billion neurons within our brain, and those form around 100 trillion synaptic connections. So these are incredibly complex networks of interacting parts that are plastic and change over time. And there's a lot of kind of regulation and control systems that are built into our brain to create these complex functions, both sensation, motor control, and thought. And it's when there's damage and disease that happens to these aspects of our nervous system that there's opportunities to intervene to treat those brain conditions and spinal cord conditions and peripheral nerve conditions with neurotechnologies. And so there's this really large design space. And the way that we oftentimes think about this is in terms of two concepts, is really intervening with technologies that can modulate or encode the nervous system function, as well as decoding or recording from the nervous system. And really this idea that Professor Ogle mentioned of responsive therapies is this idea of placing these together and creating these closed loop feedback operations. And these can be useful for studying the nervous system. And Professor Lim and I both do quite a bit of that, as developing technologies to really understand how our nervous system functions. But there's also really exciting opportunities to bring in the patient and to really tune these types of technologies to treat certain disorders of the nervous system. And so that's really the topic that we wanted to talk about with you today. So in the context of interfacing technology with the nervous system, there's a host of different ways that you can approach this. And just in the context of modulation, you can think about different forms of energy that can be used. So electrical stimulation, magnetic stimulation, optical stimulation, ultrasound stimulation, which you'll hear about later from Professor Lim. And there's also the different targets that you can think about. So there's a target for a number of brain conditions. And one of the technologies that we've developed and others have developed is this concept of deep brain stimulation for treating brain disorders, where electrodes are implanted deep within the brain, and then electrical stimulation is used to mask the symptoms that are present. There's cortical stimulation, so there's non-invasive stimulation, there's brain-machine or brain-computer interfaces. Auditory stimulation, you'll hear about that in a little bit. Retinal stimulation, we have at the University of Minnesota a number of individuals that have had retinal degeneration and have been implanted with this product from Second Sight, and it provides electrical stimulation of retinal cells, bipolar cells, to provide some visual perception. There's cranial nerve stimulation, so stimulation for obstructive sleep apnea, migraine, dry eyes, chronic pain. Vagus nerve stimulation, you'll hear about that in a little bit from Professor Lim, used for treating epilepsy, depression, obesity, and inflammation. Baroreceptor stimulation for hypertension. Phrenic nerve stimulation to control respiration. Spinal cord stimulation, there's Professor Nedoff in our department and colleagues with neurosurgery are leading a large clinical trial on treating or allowing individuals that have spinal cord injury to regain lower extremity function. It's really amazing, and you may have seen news reports on that work. Perineal stimulation for foot drop, and then a host of pelvic floor stimulation approaches through urology. So the University of Minnesota, we in the biomedical engineering and our collaborators and colleagues at the University of Minnesota are touching on a lot of these different topics, and that's really an exciting space to be in especially for our students who are engaged in this type of research. So I'm going to briefly mention Parkinson's disease and essential tremor, and then Professor Lim is going to be talking about tinnitus and inflammation in the context of interfacing technology with the nervous system. So to begin with, first off, Parkinson's disease. Many of you may know people with Parkinson's disease. My grandma had Parkinson's disease, and that's one of the reasons why I spend a lot of time focusing on Parkinson's disease and trying to understand it better. So Parkinson's disease, it manifests, there's both motor symptoms, so resting tremor, muscle rigidity, slowness of movement, as well as non-motor features. And oftentimes these non-motor features appear earlier in the disease process. And one of the things that we've learned over the years is that while the primary therapy for treating Parkinson's disease is treatment with this a drug that replaces dopamine within our brain, there's a host of other pathways that are involved in our brain. And I'm going to use pathways in the sense of thinking about how one set of neurons connects with another set of neurons within the brain. So oftentimes typically Parkinson's disease starts very deep within our brainstem, and it degenerates first in the regions that are connected with our olfactory system, so our sense of smell, and then our system that's connected to our gut. And so it's kind of interesting in that concept in terms of environmental factors that are involved, but there's certainly other genetic factors that could be involved as well. And the interesting thing with Parkinson's disease is it's progressive. And so while it starts deep within the brainstem, it then shifts farther up through our brainstem and into our larger cortical areas. And so that really brings in these different stages of Parkinson's disease. And it's really not until we see degeneration up in this area, if you can see where my cursor is, up where this arrowhead is, that we actually see symptoms. So there's certainly some non-motor symptoms that can be early indications, autonomic disorders, REM sleep disorders, olfactory impairment, but it's really not until these areas degenerate that we actually see the motor features. And that's when individuals oftentimes go into the clinic to gain treatment. And so one of the big goals is to try and figure out how we can better identify in this pre-symptomatic phase individuals that may have Parkinson's disease. But it's also in the symptomatic phase, how do we provide individuals ability to reclaim control over a lot of their lost function. And so one of those tools is called deep brain stimulation. And deep brain stimulation is a technology that's really been around since the 1950s, 1960s. And it was pioneered by a number of neurosurgeons that found that when going in and lesioning an area, which can create, so a small brain lesion can actually treat Parkinson's disease. They were trying to target that area first with electrical stimulation. And they found that if they stimulated at low frequencies, so low pulsatile stimulations, they could make the individual worse in terms of their symptoms. And by stimulating at high frequencies, they could actually improve the symptoms. And so kind of fast forward now to kind of present day, and there's a number of medical device manufacturers in the Twin Cities area that design and develop these technologies for using an implantable pulse generator that sends stimuli up and into the brain through these leads of electrodes. And so this is kind of what it looks like. Here's an MRI of an individual that was implanted with a DBS lead. So you can see the kind of artifact of that lead through the MRI. And if you take a close-up view, there's this region that's oftentimes used to stimulate in Parkinson's disease called the subthalamic nucleus. And so this STN target is one that I'm going to be talking about in a little bit. And so you can see this was an individual that came into our clinic and agreed to have his video taken. And so you can see in the context of Parkinson's disease, the slow movements and the rigidity and the difficulty with gait. And here he is after his deep brain stimulation surgery. So it's quite remarkable in how this therapy can work. But one of the challenges that we oftentimes face is how to program or figure out the stimulation settings to get these best responses. And it varies from person to person. And as was mentioned kind of in the context of responsive therapies, it very much varies from day to day and minute to minute in terms of symptom severity. So that's really the goal, is really to understand how to stimulate to treat each symptom at each moment in time. So one of the challenges with all of this is that in addition to stimulating to achieve therapy, there's also ways that if you stimulate, you can induce side effects. And some of the side effects that you can get with stimulation are overt, like a muscle contraction. And so here's an example of that. So here's our subthalamic nucleus target, and here's a lead of electrodes. So there's electrodes down below, and then as we steer the current around our lead, we can either stimulate or avoid this pathway. And so this pathway is one that goes from our motor cortex down into our spinal cord. And when you stimulate it, you can get strong muscle contractions, which are side effects. And so we want to avoid those, and we do in the clinic. And so one of the challenges with delivering DBS therapy is trying to maximize that therapeutic effect, but also trying to minimize the presence of any type of side effect. So some other side effects that have come about, some are really interesting. So in one case, there was a computer programmer that had Parkinson's disease and came into our clinic and had trouble typing on a keyboard due to Parkinson's disease. And through stimulation, we were able to relieve that. But by stimulating too large of an extent in the subthalamic nucleus, we actually caused a change where when you think about typing on a keyboard, you probably don't think about the actual keys that you're typing. And so that kind of automated behavior, we were preventing him from doing. And so he had to think about kind of each individual key that he had to type. And so what we did then is we went back we imaged sort of where his lead was located and then adapted the stimulation so that it was more focused, and that side effect was resolved. And so it's kind of thinking through these processes of trying to automate stimulation and thinking through how to sculpt it to the individual. And that really led us to thinking about these patient-specific models. And what we've done is collaborated with some great people in our Center for Magnetic Resonance Research to take high-field MRI imaging and perform segmentation and tractography and building these conductivity models to predict, one, how electrical stimulation propagates through a lead, and then two, how to predict how those potentials, tissue potentials, then interface with all of these different pathways. And what we use is a great tool at the University of Minnesota, the Minnesota Supercomputing Institute, to run these detailed computational models of thousands of different neurons across these different stimulation settings for each patient. And we can use these models, equivalent circuit models, to predict how current then flows through these individual nerves or axons or neurons, and then use that information to determine what stimulation setting actually, what it would do to the surrounding tissue. But that becomes challenging because you have these supercomputers running nonstop trying to solve all of these problems and simulate all these different solutions. So we thought, well, maybe there's an easier way to approach this. And really the goal is to figure out what is the electrode configuration that can most efficiently and effectively and selectively modulate these pathways of interest. So there's a lot of different optimization theory and machine learning approaches that you can take to solve this problem. How do you best stimulate this one pathway without stimulating this other pathway so that you get the robust therapy without having the side effect? And so one of the approaches that we've taken a whole bunch of approaches, but the one I wanted to show you today is it's called particle swarm optimization. It's a technique that was developed kind of late 90s, early 2000s, and it's a non-convex approach. And the idea is that there's probably a lot of very good settings, but trying to find that best setting for the individual is a challenge. And so honeybees solve this problem, and honeybees do this by each one going out and finding its best food source, but then the honeybees have a social component where they can communicate with each other to find their best food sources. And so that's really what we've done, is we've taken these bees, or you can think of these particles, or you could even think of these as neurologists trying to figure out what the best setting would be. And each particle then explores this very vast parameter space of different stimulation parameters. And it updates kind of from one time step to another to find the result. So here's an example of that. Here's our subthalamic nucleus again. Here's a lead placement from one of the patients that was implanted. And here are all the different pathways, at least that we've modeled. And so we've modeled up to 13 different pathways, 13-dimensional space, and all of the different stimulation parameters that could be achieved with that. So what ends up happening is these particles explore the parameter space and then eventually converge onto a solution, and then that's something that we can evaluate. So another approach is Bayesian adaptive dual control, and that's kind of thinking about things in time, and that's where you can record brain activity and then stimulate through an electrode to see how that brain activity is changed by stimulation. And so Logan Grotto, in collaboration with Tay Netoff, we've done this study where we've looked at using this kind of autonomous programming approach to sample brain oscillations in real time and then adapt sort of stimulation settings based on that approach. So just in the last couple minutes or so, just a quick piece on essential tremor. So while we were using we've used adaptive controllers for DBS therapy. There's also adaptive controllers that are intrinsic to our brain, and essential tremor is one example of that. So our cerebellum controls a lot of our movement, and disorder in the cerebellum is known to cause essential tremor. So it's this kind of swelling of neurons called Purkinje cells and pruning of their synapses that lead to postural and action tremor. And so deep brain stimulation can also be used in this context for stimulation of these different targets. So we have currently a clinical trial going on, and that clinical trial is using our machine learning approaches to try and better target a lot of these sub-targets for treating essential tremor. And so if you're interested in that, there's a clinicaltrials.gov website that you can go to see that. So the last thing in terms of essential tremor and responsive therapies is that, DBS can certainly be used in this context of responsive therapies, that you could take a peripheral sensor, an extraneural implant, or even neural activity from the deep brain stimulation lead to close the loop. And these are really active pieces of research that are going on at the University of Minnesota and elsewhere. But in the meantime, there's actually some really neat technologies that are in a form of a responsive approach. And so one of those is by Verily and Google, and it's a spoon that takes hand tremor and adapts it to counteract that at the spoon end so that individuals are able to eat without spilling food. And another one is by Health, and they have this on-demand peripheral nerve stimulation. So when tremor happens, there's a small electrical current that can pass through. So there's really exciting, I think, work kind of in this responsive therapy space, both in terms of implants as well as in terms of wearables and non-invasive approaches. So just to recap, there's patient-specific computational models can be really useful for identifying pathways that are involved, and machine learning can be really useful in the context of trying to target those pathways more effectively. And really where I think a lot of work is going to be done, and probably work that many of you did when you were at the University of Minnesota, is taking classes in like control engineering and electrical engineering and biomedical engineering, and really harnessing those approaches to create these on-demand responsive therapies. So there's a great number of people that work on this, and really it's a team effort, a great team effort with Jerry Vitek leading a lot of the work in our neurology clinic. So thank you. And I think many of the students, I think I saw Caitlin on, and I saw Oscar, and I think I saw Nasser. I'm pleased that all these students have contributed to a lot of this work. So I think I'll stop for there and maybe pass it off to Hubert. And so Professor Lim has been doing some really exciting work, just pioneering work, using a range of different neuromodulation approaches to treat everything from the things in the auditory system all the way to COVID-19. So I'll pass it on to you, Hubert.
Okay, thank you. Can everyone hear me okay? Is this good?
You're good. And just
Okay.
as you get your slideshow running, I just want to remind people that if you have any questions for either of the professors, please go ahead and type them into the Q&A feature, and we will address however many we can at the end. Thank you.
Okay, well, thank you. So Professor Johnson, thank you for the introduction, and also covering a lot of the base background for neuromodulation in the brain. It helps my talk go in more smoothly. My job is to talk about tinnitus, rheumatoid arthritis, and COVID-19. Originally it was 10 minutes. I think I have 15 minutes, but I'll make it to 12.5 minutes because I see the time is running out a little bit. Of course, there was a lot of people involved with this research. I'll just say, if you're interested, soniclab.umn.edu for a lot of the hard work done by the lab personnel that's listed there, and of course the funding sources. And I have some disclosures. I do have equity and chief scientific officer of 2 companies that I will mention today. So I do need to show this slide, and then we can move on. Before I get into the research, I did want to share some personal interesting fact about myself. I've always felt that I've been destined to be in Minnesota and do biomedical engineering research. Many people don't know this, but I was born in 1978 in June in Southern California. And of course, my mother was pregnant with me before then, and my father was trying to figure out what to name his son. Around that time, a famous person in Minnesota, Hubert H. Humphrey, had passed away in January 1978, and my father decided that this would be a popular and powerful name for his Asian, South Korean American son to be named. Little did I know that I would never meet another Hubert in my life, let alone an Asian Hubert. But somehow subconsciously, I feel this has brought me living abroad in multiple states and countries, somehow ended up in Minnesota. And so if there is someone named Hubert on this Zoom call, please give a shout out at the end. I'd love to meet you. Okay, so jumping into the topics, tinnitus or tinnitus, either way, many of you may know about it, is called ringing in the ears. Basically, it's sound, tonal type sounds, noise-like sounds that are being heard really in the brain, but there's no input coming into the ears. And it can be quite bothersome and debilitating for individuals, as you can imagine. And surprisingly, it's a huge unmet need still. 10 to 15% of the population are affected by it, yet no clinically validated drug or device treatment yet. So thinking about how we could go about treating tinnitus, you have to kind of understand a little bit of how it manifests itself in the brain or is caused. And one view of it, and I think majority of people have a consensus towards this, is that many individuals have hearing loss or some degradation in their hearing in the peripheral system. And as a result, which isn't too surprising, the brain will try to compensate for that, and it will upregulate the brain. Think of it as increasing the volume in the brain, in the auditory brain. But in some individuals, not fully understood yet, how, the brain overcompensates, and this leads to the phantom perception, the tinnitus. So when you think about it in a simplified sense, of course, this is much simplified than really what's going on. One way to target or treat tinnitus is basically to suppress that hyperactivity, to calm down that hyper activation or synchronous patterns that are happening in the brain. Now, you could do that, as Professor Johnson pointed out, some very fascinating and encouraging methods of using deep brain implants. Another way is to try to target it in a non-invasive way. So when I came to University of Minnesota in 2009, I was on a mission to find a non-invasive way that we could access those brain centers that could then treat tinnitus. And in one sense, you could play sounds and you can activate, if you can see my mouse here, different brain centers going up the sending pathway to the auditory cortex where perception happens. What we discovered in my lab over the last decade, but predominantly from 2009 to 2015, is that you can electrically stimulate different body regions. So the head, the face, the ear, the neck, the tongue, the body, and you can combine it with different types of sounds, with different types of temporal patterns, and drive changes in the brain, in the auditory brain, over a long-term scale. And we investigated this in many different parameters and conditions in a guinea pig animal model that led to promising results. So from that research, we discovered two key things. One is that you could electrically stimulate the ear and drive some of the strongest changes in the brain relevant for tinnitus that we discovered. And then the other is that you could electrically stimulate the tongue. And so both of these have been a direction that my lab and my interests have been directed towards. So in terms of electrical ear stimulation, we have been running different human studies, pilot studies at University of Minnesota, and we have obtained encouraging results. This is still in the process of being analyzed, but we do hope to share that in the upcoming year as a publication with my lead investigator, Yezi Mesfin, who's been overseeing those studies. On a larger scale, for the electrical tongue stimulation, I was fortunate to come across a company called Neuromod Devices, and they had been around since 2010 in Dublin, Ireland. We started to interact, and we realized that we were finding consistent results in using electrical stimulation of the tongue with sound to treat tinnitus. So I joined together with them as their chief scientific officer, and we decided to work together to really push forward a rigorous scientific and clinical program with large-scale clinical trials. And so what you see here is the device Linear, and it has a paddle with electrodes on it to stimulate the tongue surface that's then combined with sound stimulation. And the idea is to use this 30 minutes to 1 hour a day from 6 to 12 weeks, which was the design of the clinical studies. And what we found evaluating in 517 tinnitus patients is that within 6 to 12 weeks, we could significantly reduce the tinnitus symptoms. And surprisingly, and quite encouragingly, was that these effects could last out to 12 months post-treatment. So we are excited about these results. We're working hard to commercialize and bring the device to the US. It's available in Europe, but now moving to seek FDA approval and bring it to the US. And we have a series of publications that will be coming out in the upcoming year to 2 years. So shifting gears now to rheumatoid arthritis. Many of you are familiar with arthritis and the swelling and the tenderness in the joints. There are different types of arthritis, and rheumatoid arthritis is characterized by what is known as an autoimmune disorder. So think of it as your immune system is hyperactive, and there's too much inflammation happening in your body that is going out of control, that your body is not properly able to control that. And different links have been to genetic predisposition, as well as combinations with environmental, lifestyle, and some triggered events that cause the cascade of this happening. It is quite reasonably treated well with drugs, but of course drugs do have side effects, high costs, and there are still many individuals who don't fully benefit from these drugs. So one of the things, as Dr. Johnson pointed out, the brain has so many different connections throughout the body and different ways we can access that for various health conditions. And one of the networks that is quite exciting to me and still has a lot to be learned in the bridging of the field of the brain world and the immune world is how the brain nervous system interacts with different end organs involved with inflammation of the immune system. And in this case, you see to the right, you have the brain that connects through the vagus nerve all the way down to the spleen. And the spleen then can control the immune system or inflammation in your body. And what people have found before our group got into this field is that you can electrically stimulate the vagus nerve, and this can then modulate the spleen and suppress down the inflammation. Now, this is encouraging, but we thought the vagus nerve is connected to so many different pathways in the body, even the heart, that we thought maybe we could access more locally the spleen itself. And so we ran experiments in mice where we applied ultrasound to the spleen, and we're able to treat their inflammatory arthritis by reducing the hyperinflammation that occurs due to arthritis. So due to those promising results, we were able to start a clinical trial that's ongoing now. It's really led by my clinical collaborator, Dr. Eric Peterson. And in that study, we use a commercially available device. However, it wasn't fully optimized for the spleen and for this application for inflammation. So we have been fortunate to start up a company called Second Wave Systems in Minnesota, the Twin Cities, where we are developing portable wearable phased array technologies that can be applied over this rib area to deliver targeting of ultrasound energy to the spleen. So on the final topic here, just a few slides, is COVID-19. And as Dean Kaveh had mentioned already, I mean, this is in our lives now, and it's been challenging. So I'm sure many of you have read up on what's going on with COVID-19. You can think of it as it has to enter your body, and one key way this virus enters your body is through the respiratory tract into the lungs. You can think of it as, the virus has a key and it needs to unlock, find the locks that it can, unlock and open the door and get into the house or the cells, where then it causes spreading and damage. One of the key things, though, however, is that as the body's trying to heal itself, or whether you use therapeutics, the inflammation starts to escalate, and it can lead to what's called a cytokine storm, where you have an excess of these inflammatory molecules called cytokines. And that leads to various symptoms, but also organ failure and, unfortunately, mortality. So what we realized with a lot of the recent research coming out is that many of the inflammatory molecules that are upregulated or increased from SARS-CoV-2 or COVID-19 is actually the ones that we are observing in our rheumatoid arthritis study, where we applied ultrasound to those individuals and we characterized their blood and genetic makeup, and we are able to show that we could suppress similar types of inflammatory molecules. And so really the question becomes then, can we apply ultrasound to the spleen and suppress or calm this cytokine storm to give the body enough time and enough healthy state to tackle and eliminate this virus? And I do want to give acknowledgement to the Department of Defense and DARPA, because they are funding a clinical trial to pursue this here at University of Minnesota. So with the final slide, I know we're running out of time here. The whole idea of these treatments is that we are trying to find parameters that we can intervene, apply energy to the body and the brain to treat these different conditions. But really the holy grail, because of the diversity, the variability we see in these different conditions across individuals and the varying timescale of how it manifests itself or sustains itself in the body, we do really need to find ways to take information from the body, whether we have sensors that pick up the nervous system patterns, or we have non-invasive molecular sensors. Those then can then feed back to drive the energy stimulation. And we do have at the University of Minnesota now a newly built lab to investigate this that's supported by generously by the College of Science and Engineering and the medical school called Test Lab. And we hope to push these types of responsive therapies in this lab as we move to the next era of energy stimulation and treatment. So with that, I'll stop there and we'll take any questions. So thank you for your time.
There we go. Looks like we're all back. You can continue to submit questions again using Q&A, and we'll get to as many as we possibly can. And again, the questions that we'll be able to address today are for Professor Lim and Professor Johnson, but there are a few others that we will absolutely follow up with you about after today's session. So I will start off by reminding you all that I am not the expert, so professors, you feel free to define the scope of what you can answer. So I'm not sure if all of these questions fall within the scope of your research, but I will ask them anyway, and you can let us all know. But first off, Professor Lim, is there any evidence to suggest that diet can reduce inflammation to help reduce the symptoms of rheumatoid arthritis?
I don't want to say definitively, but I will say that my interest is along those lines partially. I do believe that the health of the body, its physical health, but also mental health, all of these factors contribute to the immune system. And so there are studies that are coming out that, stress level, emotional level, various health states can affect your inflammation level as well. And so I do believe that a lot of those factors, although we don't know yet know how, but being able to control or adjust those factors, as well as using other therapeutic approaches like what we presented today, I think a combination of those factors is really the future direction of how we're going to find a comprehensive way to treat these different conditions. So I'll leave it at that because it is still a new frontier to explore further.
Wonderful, thank you. I will have Professor Johnson start with this question, but I think that both of you could probably address it. Someone is on this call who participated in a 2018-2019 study, and he is wondering what your next studies must be. And I think you kind of alluded to some of those, but perhaps you can tell us what you're most excited about.
Was that to me or was that to Professor Johnson?
Professor Johnson, why don't you take that first, and then Professor Lim, you can go after.
Sure, yeah, I'm really excited about just the idea of using machine learning tools to help. Sometimes we're faced in sort of clinical situations with either so much data that we're trying to make sense of, or there's just limited amounts of time to make decisions. And I'm really excited about the idea of using machine learning and perhaps artificial intelligence to be sort of a complement to kind of existing clinical procedures. So we have a couple clinical trials kind of exploring this idea of artificial intelligence in healthcare, and I think that'll be a really neat opportunity going forward. The Medical Devices Center at the University of Minnesota is doing some phenomenal work across the board in translating all sorts of different technologies, and so I think there'll be a lot of really exciting work partnerships with the medical device center and the hospital system and the biomedical engineering department going forward.
So I would just echo what Professor Johnson said about artificial intelligence and machine learning. I do believe, these health conditions are quite complex, and we do need to apply these more sophisticated techniques. I know this might sound far off, but if we can develop more sensors and non-invasive approaches, I would like to approach it on larger scale, even to benefit from the machine learning, where individuals at home, we could ship them devices, or they would be purchasing it even from Amazon.com, and they could get these devices, and we could really explore lots of information and different kind of methods to collect that kind of data. Because I do think, we try to do things in animal research and we try to extrapolate to what it means in humans, but we do need a lot more human data to really tackle these complicated health conditions. And so moving towards that direction and kind of big data, big combinations of lots of people contributing, I think this would be the future where we can really tackle these tough problems.
All right. Professor Lim, is there any way to use ultrasound to directly reduce inflammation of the lungs? And that question is specific to COVID-19.
So I don't want to say no, because so I believe that ultrasound is modulating cells, modulating neural cells, nervous system cells, but also immune cells. So I do believe that where we can target the ultrasound, like in the lungs, we could modulate then cells there as well. Obviously, the big question is what parameters and how to modulate so that we cause the cells to calm down their inflammation and also to target the right cells. And so this is something that, of course, is challenging to do, but definitely a possibility for targeting the lung directly.
Professor Johnson, I'll have you take this one. Ken is wondering if you usually partner with biomedical firms on your research.
Yeah, I think that's one of the really exciting things of a lot of the research that's going on in our department is a lot of partnerships with the medical device field or medical tech field. And there's a lot of great companies around the area, and I see quite a few people that I recognize from a number of those companies who are alumni of the University of Minnesota and so it's a great opportunity, I think, one, to really see a lot of the technology translated, and then two, I think it's a great opportunity for students to gain that kind of experience working in kind of an academic industry partnership. So it's so we have a number of collaborations.
And I will let the two of you decide who should take this question, but Leonard is wondering if we have ways of knowing what our individual inflammation state is.
Good question. That is a good question. I'll leave it to be determined. That's a tough question. It's something that we are also hoping to discover, to then be able to better treat and better make personalized stimulation patterns for each individual. I mean, I guess I could say, we collect blood samples, do some genetic analysis and different biomarkers, but I think it'll also incorporate, neural aspects that we need to extract. So it'll be a combination of information that will come together to get like a fingerprint for each individual. But very good question. Don't have an answer now. Maybe in 5-10 years, I will.
And it looks like we have time for one more question. And I will say, as I said before, we have so many wonderful questions that were submitted, and I apologize that we aren't able to get to all of them. My goal has been to take some of the more general questions that aren't specific to certain conditions, because there are a lot of very specific questions. And I wish we had another hour, but I'm sure people have places to be. So to be continued, and we'll get back to you as much as we can to address the things that we can't right now. But interesting question. And I guess you can both take this, and as you're answering, feel free to throw in any final comments that you'd like to share with the audience. The question is there a difference between genders and how often neurodegenerative diseases occur? for example, Parkinson's essential tremor, or is the genetic component gender neutral?
Yeah, that's a great question, and it's actually a fundamental question to a lot of science because it's so important in research to make sure that we have gender balances in the research that we do to really understand how different therapies and different brain conditions or other conditions kind of affect may affect different genders. Parkinson's disease tends to be slightly skewed towards a more male population. And then essential tremor tends to be skewed a little bit more towards a female population, just in general. But in terms of overall kind of therapeutic approaches, I haven't seen anything that would sort of differentiate those two, at least from the standpoint of neuromodulation. So it's more of a prevalence, maybe, rather than sort of a treatment. But Professor Lim?
Yeah, I think there's not enough. So we do know that gender does affect different physiological and neural and immune patterns in the body. That is clear. And even, when we anesthetize animals, we'll have different levels of anesthesia effects depending on gender. As to how neuromodulation or energy stimulation of the body affects in different ways, there's just so little, there's very little done to characterize that. But I would predict that when studies are being done, and we are pushing that way because through federal funding, we do need to look at these differences, we will see that there probably will be some differences in which parameters and which conditions are more effective based on gender. At least that's what I hypothesize.
Okay. With that, I'm going to share my screen one more time so you can see that we're out of time for today, but we do have another lunch and learn opportunity coming up for you all next Thursday, August 27th, and that is hosted by the Department of Electrical and Computer Engineering. And the keynote speaker for that presentation will be Professor Bethany Stadler, who has been with the university for many, many years and done some wonderful research as well. And she will be talking about her team's use of magnetic nanowire to drive advancements in human health. And you can register for that event with the URL that's on the screen, z.umn.edu/ECE Lunch Learn 2020. Don't worry, you don't have to remember that. You don't have to write it down. It will be included in the follow-up email. We'll send a link. Again, another complimentary opportunity, and we will be offering these regularly throughout the fall. We have probably another 3 coming up in the fall, and we'll do it throughout the year as well, and hopefully continue to feature numerous other departments. So thank you again for joining us today. Thank you for all of our wonderful presenters. We appreciate you all being here. And again, that follow-up email will be coming in the next couple of days, and it will have the link to today's recording that you're welcome to share, and a few other event opportunities that are coming up. So Moss, would you like to say your final goodbye and we'll sign off for the day?
Well, what can I say? I'm always amazed to hear what our colleagues are doing. I know bits and pieces of it, but we call these lunch and learn, and it sure is learn for me. Amazing, amazing work going on in your college. Thank you so much for joining us, and hope to see you at the next event and many more. Stay well.